The Story of Lactose and Lactate, Lovers to Enemies, and Infamous Gene Culture Coevolution Breakdown by Katie, Taya, and Daisy.
So first, we're going to start by explaining natural selection. Natural selection is a mechanism of biological evolution where organisms' descendants adapt with modification to fit specific environmental needs. This was first coined by Charles Darwin when he observed various finches in the Galapagos Islands in 1835. He noticed that different beaks were suited to different environmental niches on the island and would fluctuate in the population depending on how successful each beak type was in collecting food. If one beak shape was no longer successful within the environment, a different beak shape would become more prevalent in the society due to the finches with the unsuccessful beak shapes dying off because they don't have the selective advantage of having the better shaped beak. It's also important to note that just because a beak shape is not necessarily successful within the environment, that it won't not appear in the environment. These shapes will naturally appear just due to natural variation within a population. So now we'll look at how this evolutionary mechanism can be applied to humans in our biological evolutionary history. One of the most famous examples that we can see is lactase persistence. Lactase persistence is part of a natural process where mammals have an enzyme called lactate that is used for digesting milk that is prevalent in childhood, but as the mammal grows into adulthood, the lactate enzyme gets phased out as they no longer need to digest milk sugar lactose. However, as you may have noticed already, there are many individuals within our society that do consume and digest milk still. So how can this be? Well, it's due specifically to genes. A gene is a physical or biological trait inherited by offspring from a parent in the offspring's DNA. An example of this would be the beak shapes that we talked about in the previous slide. Genes are made up of alleles, which are specific copies of a gene.
What is lactose? Lactose is a carbohydrate found in mammal's milk. Lactose is also an enzyme. Lactose is a disaccharide, which is a sugar that can be broken down into two smaller sugars known as glucose and galactose. Glucose and galactose are monosaccharides. Lactose can be split into these two monosaccharides when an enzyme known as lactase is produced. How does the lactase enzyme function in the small intestine? The lactase is an enzyme that that resides in the small intestine of mammals. Enzymes are a complex protein that is a driver for chemical processes within the body. Enzymes are transported by protein molecules known as polypeptides. As lactase interacts with lactose, lactase breaks down the lactose to be easily streamlined into the body. To have or not to have lactase. So lactase will be the pink little diamond and lactose will be the circle with the cross in it. So lactose, when it hits the small intestine, it is metabolized by lactase and dispersed throughout the body for energy. If there is no lactase, lactose will build up into a series of unpleasant symptoms such as cramps, diarrhea, and flatulence. These symptoms are from having lactose intolerance. The adventure of lactase persistence. As lactose travels around the world, he finds himself in fluctuation across populations. Lactose has two friends, glucose and galactose, that we will call GG. Lactose and GG hold a strong relationship that is consistently being preyed upon by lactase. Lactase persists to split these fellows apart in order to try to swindle lactose's love, but often her absence lets them roam free. Adventure continued. If lactase and lactose fall in love, what may happen to GG? What if lactase and lactose only see each other secretly in different parts of the world? What might that tell us about the relationship and the populations? If lactase persisted to conquer splitting up lactose and his friends GG across the world, would there be more or less lactose intolerance? What are some factors that could let lactose and lactase have a tumultuous relationship? If lactase died, what may happen to lactose and GG? Here are those answers. So, on one, GG would be split apart but may be together in certain populations. 2, their secret relationship might mean that the culture around lactase is supported. 3, there would be less lactose intolerance because lactase would succeed at breaking apart the lactose in GG. 4, lactase and lactose can have a battle between each other if there are less or more support from the populations. Variations may occur. 5, lactose and GG may work together throughout populations, but lactase persistence may occur throughout populations due to lactase being around in the beginning.
Historical context. Lactose persistent genes are seen in higher frequencies in populations that have a high incorporation of milk in their diet. These are the populations that would develop lactose persistence via natural selection. Those with the genes to digest lactose into adulthood had better nutrition and lived longer and reproduced more successfully than those without. Believed to have first evolved independently in pastoralist societies in the Middle East for people who kept livestock but did not farm.
Another evolutionary force that can impact allele frequency is genetic drift, which is a change in the allele frequency by chance rather than being overtly selectively advantageous within the population. So an example of this can be seen with the spread of lactase persistence throughout Europe. Europe had pastoralist societies around 6,000 years ago, but it was not its main source of subsistence. DNA archaeological evidence proves that the populations did not have the mutated T13910 allele frequency commonly noticed until 4,000 years ago, 2,000 years after pastoralist societies had been introduced already. This delay suggests that milk consumption was most likely limited to children at first, as they could still digest the lactose, and adults most likely just tolerated the side effects but still consumed lactase.
Natural selection in human culture. Lactose persistence developed due to cultural changes seen within human societies and increased reliance on specific subsistence sources or simply taste preferences. The evolution of the gene is not only an example of biological evolutionary mechanisms, but also cultural evolution, making it a unique example of coevolution.
Evolutionary forces check. We have already discussed 3 of the 5 main evolutionary forces: natural selection, mutation, and genetic drift. The other 2 include non-random mating, where mate selection is influenced by phenotypic expressions and underlying genotypic differences. Gene flow is a migration into or out of a population that can influence allele frequency within each population. Alrighty, using what we discussed, link the evolutionary force to the situation.
How do genes function? DNA is transcribed into mRNA, and then tRNA, which is bound to an amino acid, carries an anticodon that binds only to the mRNA with that codon. Ribosomes help detach the amino acid from the tRNA and glues the amino acid into a protein chain. DNA controls whether a gene is repressed or expressed by affecting whether or not the proteins that control transcription bind to the DNA. These proteins then control phenotype and variation, which means DNA is subject to natural selection.
Applying the nature of genes. So there are 2 types of mutations that can occur within a gene. A point mutation, which is a change within a gene where one base pair that consists of single nucleotides within the DNA strand is altered, and a frame shift mutation, which is an insertion or deletion of a single nucleotide within an amino acid sequence, completely changing the order of the sequence. So now we'll talk about this question. The mutation responsible for lactase persistence is called a SNP, or a single nucleotide polymorphism. The allele has a base pair that is mutated from C to T and inversely G to A. What type of mutation would this be considered as? it would be considered a point mutation.
All right, from what you know, we're going to put it to the test. What is with all this DNA? Please put the order in which DNA codes for protein synthesis. What type of information is represented in DNA? Why does DNA need to self-replicate? Adenine binds to blank, whereas guanine binds to blank. Where is the location of a gene in DNA?
Time for some milk. Can you handle it? DNA is a string of bases that are attached to one another and rolled up super tight. They come in pairs. Thymine is in love with adenine and cytosine is in love with guanine. For example, CCGTGCTAG would pair with its corresponding part and become a part of a string of DNA. But during transcription, adenine cheats on thymine with uracil, meaning this sequence will look very different. Uracil then finds out and cheats on adenine with thymine. Also, but adenine and thymine always work it out and get back together in the end. Within each strand is the means to make amino acids, the building blocks of protein, which are paired together in a sequence. For example, if you use the first 3 letters of the parent strand in the above CCG, codes for proline according to the codon usage chart. For this activity, try to transcribe the codon for lactose gene. Hint, it's easier to translate and transcribe in 3s. Alright, as you can see here, every A now corresponds with a U, and so forth. Using the diagram to the left, translate the new set of codons, which we just transcribed in the previous slide, into the amino acid chart. For example, AUG equals MET, which is an acronym for methionine. First, go to the left and put the first base in, then go to the top and put the second, and then the third base on the side. Make sure they're all lined up in the same column and row, and that'll give you the amino acid it codes for. Here are the answers for the codon amino acid chart. Lactose persistent punnets. Lactose persistent is a recessive trait. Here we are going to use Punnett squares to determine what the probability is for an offspring to retain the recessive trait if one parent is heterozygous for the gene and the other is homozygous. Let's take a moment to figure it out. As you can see, 2 sets of alleles only produce 4 chances for an offspring to have those alleles. In this case, the offspring to have the recessive gene, there are only 2 of 4 chances that actually have the gene. The probability is 50%.
There are other forces that can impact allele frequency besides evolutionary forces. This includes variation, heritability, and reproductive success. Using the following definitions, assign them to the correlating examples.
The Hardy-Weinberg equilibrium is an equation that assumes that there are no gene mutations, no gene flow, random mating is occurring, there's no genetic drift, and no natural selection changing allele frequencies. Let's pretend there's a population of 1,000 individuals on an island. 100 of them are lactose persistent and the rest are lactose intolerant, or impersistent. We want to know the frequency of alleles present within our population. Use these equations to solve the problem. Given that P equals the frequency of dominant alleles, Q is the frequency of recessive alleles, P squared is the frequency of homozygous dominant individuals, Q squared is the frequency of homozygous recessive individuals, and 2PQ equals the frequency of heterozygous dominant individuals. Reminder, lactose impersistent individuals are homozygous recessive. Now let's solve. If there's 1,000 individuals, that means there are 2,000 alleles, because every individual has 2,000 alleles. 160 people are lactose persistent, which means there are 320 alleles. 840 of them are lactose impersistent. So if we take that and we multiply it, and what we're looking for right now is Q, and if Q is the frequency of recessive alleles, that means we need to divide 320 by 2,000. We get Q equals 0.4, and if we're using the equation at the top, P plus Q equals 1, then that means P equals 0.6. So from here, we can take those numbers and plug them into the equation, and we get Q squared equals 0.16, P squared equals 0.36, and 2PQ equals 0.48. Here we can see that there are 480 individuals that are heterozygous, 360 individuals are homozygous dominant, and 160 individuals are homozygous recessive. And that's how you do the Hardy-Weinberg equilibrium. Thanks for watching. Bye.